In this recording, I'd like to discuss the physical control of microbes. This information is from Chapter 11. When we talk about controlling microbes, we are referring to methods to destroy or reduce microbes in a given area. These can either be physical methods or chemical methods. First, we have to understand a little bit about the relative resistance of microbes. Some are rather easy to eliminate from the environment, where others pose particular problems. Those that present the highest resistance are those organisms that produce spores. So organisms like Clostridium and Bacillus are very difficult to eradicate because of the difficulty of eradicating spores. And as you learned about endospores earlier this semester, you learned that they are quite resistant to heat, and it takes special procedures such as autoclaving in order to destroy them. Others that have moderate resistance include Pseudomonas species, Mycobacterium tuberculosis, Staphylococcus aureus, and cysts. These are the resting stages of protozoa. Those that are the easiest to eradicate include most vegetative cells. So the cells of Bacillus, for example, would be quite easy to eradicate. The spores are not. Fungal spores, envelope viruses, yeast, and the reproductive stages of protozoa called trophozoites. Some terms to be familiar with. One is sterilization. Sterilization, you know, is an absolute. So this is a process that destroys all viable microbes, including viruses and endospores. Disinfection is a process to destroy vegetative pathogens, but disinfection typically will not destroy endospores. Sanitization is any cleansing technique that mechanically removes microbes. I remember seeing a sign in Taco Bell once that was proudly displayed on the back wall that said, "We sanitize our hands." And certainly I hope they would be sanitizing your hands, because that simply means just washing your hands. And then finally, degermation. Degermation is a process that takes a little bit more effort than sanitization. A physician, for example, will degerm before donning gloves before surgery. And degermation reduces the number of microbes through a scrubbing process. Microbicidal agents are those that are going to kill. So cidal refers to kill. Bactericidal will kill bacteria, but not necessarily spores. If the product says sporicidal, that means it will destroy spores. Fungicidal and virucidal are going to destroy fungi and viruses respectively. Microbistatic agents are those that are just going to keep the organism at bay. So the organism will be unlikely to reproduce. They will be inhibited, but once the agent is removed, then the organism can go back to its life cycle. So there are bacteriostats and fungistats, no virustats because viruses are not considered living, and many agents that are used on living tissue are static in nature. When we talk about microbial death, we really do not mean that the microbe dies, but it refers that a microbe is unable to reproduce, even under optimum conditions. If an organism can't reproduce, eventually it will die, but this means loss of reproductive capability. There are a number of factors that will influence the action of antimicrobial agents. We'll see these again when we look at antibiotics as well as antiseptics and disinfectants, the chemical measures to control microbes. So first we have to know something about the number of microbes in the area that we're interested in getting rid of the microbes. So for example, will there be a high population? Will there be a lot of organisms? And also the nature of the microbes. Is this a mixed population of organisms, or are we expecting just a single organism or virus to be there? The temperature and the pH of environment is going to be important. How much of the agent must be used? Very often we think if we use a higher concentration, it's going to work better. That is not necessarily the case. The way the agent works, and finally, whether there are solvents in the environment, organic matter. By organic matter, we mean things like blood, feces, urine, vomitus. These types of things are going to affect whether or not the antiseptic, disinfectant, even the chemical method is going to work. We also want to know if there are any type of inhibitors in the environment. So it's not an easy thing to pick an antiseptic, a disinfectant, a chemical agent a physical agent to destroy microbes, we have to know a lot about the population that we're working with, including the human population, because of some of the dangers involved with these chemicals or physical methods. The cellular targets of control include the cell wall, so there will be agents or physical methods that are going to attack the cell wall of the organism. The cell membrane can be attacked. The synthetic processes, including DNA replication and RNA synthesis, DNA synthesis, and then the proteins also may be affected. When proteins are affected in the cell, we're usually talking about affecting proteins that are involved in enzymatic reactions, so metabolism stops, or proteins that are going to transport materials across the cell membrane. When these are denatured, then the cell cannot reproduce. So here we see a membrane. Surfactants are detergents. So detergents are capable of disrupting a membrane, which is made of a phospholipid bilayer. And when surfactant molecules, such as detergents, are going to disrupt the membrane, we see that the membrane becomes leaky. When proteins are targeted, the proteins can denature. Denaturation can mean complete destruction, or it can mean denaturation, but the protein can be repaired. For example, here we see an enzyme. This enzyme is going to fit a substrate, and this is the native shape or the natural shape of that enzyme. Enzymes are proteins. They fold into their native shape. If we expose enzymes to heavy metals, for example, heavy metals including gold, silver, copper, very often these are going to block the active site, and the active site then cannot admit the substrate. When the substrate does not fit into the active site of the enzyme, then the product is not made. Heat or pH change can cause a temporary denaturation of a protein. Here you can see a different shape. When the shape has changed, the substrate will not fit into the active site and the reaction will not continue. Now, sometimes pH change, if it is only minor and the organism goes back to its natural pH, then the enzyme can resume its shape. However, if heat is extreme, for example, boiling or autoclaving, this can cause complete denaturation of this protein or enzyme molecule. Extreme pH also can cause this. In this case, this protein molecule will not go back to its native conformation. It has been completely denatured. There are a number of practical concerns that we have to consider before we choose a physical method or a chemical method. First, do we have to use sterilization? So, is it necessary to sterilize what is going to come into contact with the patient or what the patient is going to come into contact with? For example, is it necessary to sterilize a bed or a gurney? Probably not. It is necessary to remove surface microbes, but sterilization is not necessary. Is the item going to be reused? If it is going to be reused and going to come in contact with another patient, then we have to consider how it can be sterilized. Can the item withstand heat, which is a typical method used for sterilization, pressure, radiation, or chemicals? For example, endoscopes. Endoscopes cannot withstand heat, so we'll have to use some other type of measure, usually chemical, in order to disinfect it. Is the method suitable? And will the agent penetrate to the necessary extent? So will we see, this is a particular problem with endoscopes, because endoscopes are going to be used, for example, in the respiratory route, for example, and they have to be decontaminated before they're used before the next patient. Will the agent that is used for decontamination, will it sterilize? Will it only remove microbes from the surface? And finally, is the method cost effective? And is it labor efficient? And finally, is it safe? So the methods of physical control that I'm going to cover in this lecture include heat, cold temperatures, desiccation, which is drying, radiation, and filtration. Heat is using hot water or steam. The mode of action of heat is going to be denaturing proteins. So the higher the temperature, the more likely the protein is to be denatured. This also will destroy, heat will also destroy membranes, and it can destroy DNA. DNA is held together by hydrogen bonds. Hydrogen bonds are broken by high temperature. So the method we most commonly use in our laboratory is sterilization. All of the media that you use will be sterilized before it comes to you, and that uses an instrument called an autoclave. An autoclave reaches 15 pounds per square inch of pressure, and that takes us to 121 degrees Celsius. The time is anywhere from 10 to 40 minutes, depending upon the load that we put into the autoclave. Pressure is necessary in order to reach 121 degrees Celsius. Steam is only at 100 degrees Celsius, so Boyle's law tells us that we can only get to higher than 100 degrees Celsius if we apply pressure. So 15 pounds per square inch is applied. This is above sea level, and this is going to sterilize the material in the autoclave. Intermittent sterilization, which you learned about earlier this semester, is unpressurized steam at 100 degrees Celsius for 30 to 60 minutes, followed by an incubation period, and then repeating the steam exposure. And this is a technique that you learned as tyndallization in Module 1. Heat is used for disinfection. It is also used for pasteurization. Pasteurization is a temperature of slightly below 100 degrees Celsius just for seconds. This is done to kill salmonella and listeria and reduce the overall microbe count, but you know that pasteurization is not a sterilization technique. It is an aseptic technique. It only disinfects. Boiling at 100 degrees Celsius for 30 minutes is commonly done after flooding or natural disasters or some problem with your water supply. This is going to destroy non-spore forming pathogens and is going to be adequate for decontaminating water. However, it is not a sterilization technique. It is a disinfection technique. Here you can see a very large autoclave. Large autoclaves are used in hospitals and research facilities. Some universities will have large enough autoclave devices like this. We actually have a tabletop model. Our tabletop model has a steam jacket, so it does not have an external source of water or an external source of steam. There's water that is around the jacket, and the water comes to a boil. Then the steam enters into the chamber. This pushes out any air that is in there. Then we apply pressure to this, so that's why we have this submarine type of door on here, so the door doesn't blow off. And then the steam is going to be pressurized. It's going to reach 121 degrees Celsius. Dry heat is going to use higher temperatures than moist heat, so it can sterilize, but typically things will take a longer time, except for incineration. Of course, incineration is going to destroy any microbes, and it's also going to burn up things, so we can't incinerate anything unless if we don't expect to recover it, then we can use incineration. We use incineration in the lab. So when you've been flaming your loops and needles, this is incineration. This is going to combust and dehydrate cells. What you see here is called a bacticinator. You're going to meet this bacticinator in lab. We are going to see the bacticinator in our laminar flow hood, our biological hood. These are expensive. They work really nicely, but they are quite expensive. It eliminates the need for an open flame, so many laboratories will use this. It consists of a ceramic core. The ceramic core gets hot enough, and then you stick the loop or the needle in there. The problem is you can't heat fix slides. A laboratory that would need to heat fix slides usually has some type of a tray that you place the slides on that will heat them to a high enough temperature that will coagulate the proteins. Dry ovens we can use at about 150 to 180 degrees Celsius, and this will coagulate proteins. Usually this takes a longer period of time. Cold temperatures are going to hold microbes at bay. That's why we have refrigerators, so you know that your milk doesn't spoil in there quickly, but the milk eventually will spoil. The microbes that are in your milk are going to be slowed, but they still will reproduce. Refrigeration is 0 to 15 degrees Celsius. Freezing is below 0 degrees Celsius, and so we can preserve food, media, and cultures. Freezing is going to be more effective when the ice crystals form and they puncture the plasma membranes. Enzymes typically are not destroyed by freezing temperatures, so they can regain their native conformation and can still work. So things will eventually spoil in the freezer as well, and very often freezer burn is going to damage food or media, whatever is placed in the freezer. Desiccation is drying, and this is the gradual removal of water from cells, and this leads to metabolic inhibition because without water, metabolism cannot continue. It's not really an effective microbial control because many cells retain their ability to grow when water is reintroduced. So you probably have some raisins in the back of your closet. You'll see that you won't have microbial contamination on them simply because there is no water for the microbes to grow. Now, fungi need less water in order to reproduce, so very often when we have dried materials like jerky, beef jerky, for example, has a lot of salt as well as no water, so we don't see microorganisms growing on them. The only possibility of a microbe most likely is going to be a fungus because fungi don't mind that salt, and fungi also don't need as much water in order to reproduce. Radiation can be used, and it is very commonly used to destroy microbes on surgical material. It has very deep penetrating power. It's going to break the DNA double helix. The covalent bonds are going to be broken. Hydrogen bonds are going to be broken. And then once the DNA is destroyed, the microbe can no longer reproduce. Gamma rays, X-rays, and cathode rays are all ionizing radiation that will destroy DNA. So we can use gamma rays commonly to sterilize medical supplies and food products. The gloves that surgeons use, for example, that are sterile, they are already packaged in a paper sheath. They are packaged in that paper sheath before they are sterilized. Once they're packaged in the paper sheath, then they are exposed to gamma rays, which will destroy any microbes that are within the packaging. The same as the Petri dishes that you use in lab. The Petri dishes are packaged, they're empty, packaged into plastic bags, and the plastic bags with the Petri dishes inside are exposed to gamma rays. That way nobody has to handle anything and package this stuff into the plastic bag and seal it up, which would cause a possible contamination. Non-ionizing radiation you learned about in Module 3, and non-ionizing radiation is UV light. Non-ionizing radiation has little penetrating power, but it can sterilize air, water, and solid surfaces. You know how UV light works. It creates the thymine dimers. This is an incorrect word here. It should say pyrimidine dimers or thymine dimers, which are going to interfere with replication of the DNA. So here we can see DNA being broken. This happens with non-ionizing radiation. With ionizing radiation. With non-ionizing radiation, you can see abnormal bonds forming between the pyrimidines, and this is going to lead to difficulty in replication for the organism. As you learned, microorganisms have repair mechanisms to get past this damage from UV light. Here again you see this image. UV light creates these pyrimidine dimers. We have two thymines next to one another. These two thymines are going to dimerize. This is going to create covalent bonds between the thymines. Here's an image of water treatment with ultraviolet light. So these are banks of ultraviolet light that can be used in a water treatment plant. Very often water treatment plants will use ultraviolet light in addition to other mechanisms, specifically chemical treatment and filtration as well. Ultraviolet lights actually can easily become fouled by biofilms, and microorganisms can grow over the surface, and this would reduce the effectiveness of the light that is sterilizing the water. So it's really only useful for small water treatment plants. Filtration can be used as well. This is physical removal of microbes by passing it through a gas or through gas or liquid through a filter. So you can see a filter down here on the bottom. The microbes are trapped on this filter. Let's see if I can get this on here for you to see it right here. So this is what we call a millipore filter. A millipore filter is made with a nylon type of a filter, and it is penetrated with ionizing radiation. That makes tiny little holes in it. You can actually purchase filters with different size holes. So if you know you only want to trap staphylococcus, for example, or bacteria, you can buy one that has a larger hole pore than one that has a smaller pore. When I used these when I was sterilizing media, and I also used these when I was trying to isolate viruses. And you can actually buy filters that have small enough holes in them that the viruses will be trapped. So heat-sensitive liquids, for example, can be filtered. Excuse me. If we cannot put it through the autoclave, filtration can be used. It's a slow process, and you need suction in order to do this because the holes are very, very small in these filters, so it does take quite a long time. We can also filter air. You probably have heard of HEPA filters, high energy particulate air filters. Your vacuum cleaner might have this. HEPA filters are very commonly used in reverse isolation rooms, and they're also used in industrial clean rooms.